Bismillah Rahman Rahim. We start the 2022 uploads for the first paper 4 that I'm going to discuss now, and I hope hopefully we should be putting on some more papers so that you can revise well for the paper 4, which is I feel the most difficult question paper in the entire biology exam. So best of luck. Now the first thing to notice in this is that the time has remained the same for the 2022 exam, but one thing has changed. There's no section B. Section B has been removed, so it's only going to be section A. There is going to be no essay questions. Now, this is the first year that you're going to have a question paper with no essay questions, which had a choice because you have to do one question out of two. So now that is now no longer available for the 2022 exam. Please, everybody remember that. I hope all of you know that. But the time is the same, but this section B has been removed. So section B no longer now is going to be, there's going to be no section B. The rule in doing the paper 4 is very simple. Number one, you read the whole question. I don't say read the whole paper, I just say read the whole question. Read the first question and see if you can do it, then do it. Otherwise, go on to the next question if you don't know anything about the chapter on the kidney. So figure 1.1 is a diagram of a kidney nephron and some of its blood vessels. So there's the marking A, B, C, D, E, F, G. Now you know A is the afferent arteriole. Why? Because it's got a wider lumen. And this has got a narrow lumen. So this is the efferent. So this is the afferent, this is the efferent arteriole. And I always tell you to remember it, E for efferent, E for exit. So efferent arteriole, and this is of course the afferent arteriole. And then of course you have the glomerulus. So C would be the Bowman's capsule, D would be the PCT, F would be the DCT, and E would be the loop of Henle, and G would be the collecting duct. Now, if you remember this, you can only do it if you know it very clearly. With reference to table 1.1, complete table 1.1. With reference to figure 1.1, complete table 1.1. Please be very careful when you're reading this. There's always some sort of a clue in this. Using the letters A to G, each letter may be used once, more than once, or not at all. So, efferent blood vessel, part of the nephron containing cells that respond to ADH, antidiuretic hormone, part of the nephron where podocyte cells are located. Part of the nephron containing cells are located in the medulla. Now, I'm going to just share a diagram with you, and then I'm going to come back to this question. Now, if you look at this diagram, I mean, I love this diagram because it's so very well explanatory, that you have to understand is that what is in the cortex. So this part is the cortex, which you have got to understand. This part is the cortex, this is the medulla. So which are the parts? Now you see the glomerulus is there. The glomerulus is here, the PCT and the DCT. Now just look at the labeling. This is the DCT, this is the PCT, and then only the loop of Henle is in the medulla. So if you look at it, this is the dividing line between the cortex and the medulla. So the glomerulus, the PCT, DCT is only the loop of Henle's, and you know some loop of Henle's are short, and those are called cortical nephrons. So these are the cortical nephrons. Why? Because they are got short loop of Henle's. And then we have the juxtamedullary glomerulus. So the juxtamedullary glomerulus is this one, which has a long loop of Henle, and this has a short loop of Henle. Now, I love this diagram because it really explains it, and if somebody has the vision to understand it, I'm sure he is going to develop a very good understanding of the whole situation and then be able to understand. And then you can see the collecting duct. Now, if you look at the label, this is the collecting tubule. Now, I'm just going to color it here. So the collecting tubule starts right from here and passes through the cortex and through the medulla. And then, of course, it's starting from here. So, junctional tubule. Now, I'm going to just remove everything. I'm going to clear this thing so you all can have it as a. Now, you can look at it and just pause the video here and have a look at it. And then we go back to the question. The diagram, which I like very much, and of course, it helps you to revise the whole chapter, especially the structure of the nephron. And you can see the vessels which are called the vasa recta. This is the ones which you need to be important. Vasa recta are the cells which are surrounding the loop of Henle. Then you can see the collecting duct, the DCT, the peritubular capillaries, the proximal convoluted tubule, the efferent arteriole, the afferent arteriole, the one which enters it, the efferent which leaves it, and the glomerulus and the Bowman's capsule. Now you can also see which parts are in the cortex and the medulla. As you can see, the efferent blood vessel was B. The part of the nephron containing cells that respond to ADH was F and G. Now what are F and G? F is the DCT and G is the collecting duct. Then where podocyte are cells are C. So the podocyte is somewhere here. So those are the cells of the podocyte which we discussed. I've discussed this in one of my videos as well. And then part of the nephron containing cells that are located in the medulla. Now, as I told you, the medulla is E and G. So this part of the medulla, this is sort of this like this. It's going to be something like this. A line passes through here. So E and G are in the medulla. This is all in the cortex, this part. This is all the cortex. And this part is all the medulla. So just revising all this was a good exercise for you to be comprehending any question on the kidney. Coming to the question on describe and explain how the cells of the PCT are adapted to carry out selective reabsorption. Now, there's a very small percentage of questions which are going to be direct questions, and you should be actually targeting those because those are the ones in which you can score well. And so you're sure to get your 20% of the marks right. So please be very careful in writing the answer to this. Is 5 marks for this, but you've got a whole lot of area to really write a lot of stuff, which is going to really get you your 5 out of 5. Now I'm just going to revise this for you. Now, this is a beautiful diagram which explains this all very, very well. And I always like this diagram a lot. And you look at all the points which are given to you, and you can see how they have shown you all these. These are the tight junctions here, which you can see here. These are the tight junctions, and very well explained. Please pause the video here and have a look at it. Number one, at the basolateral membrane, sodium is pumped into the interstitial space by the sodium potassium ATPase. That is the sodium potassium pump, which is 3 sodium out and 2 potassium in. And I made the mnemonic for that is Nokia, which I hope you remember from my videos, Nokia. So this is 3 sodium out and 2 potassium in always. So I know, of course, we don't use the Nokia phones anymore, but still it's a nice way to remember it. 3 sodium out, 2 potassium in always. So at the basolateral membrane, you have this. You can see this here. This is 1 here, which we are talking about. Then 2 downhill sodium entry into the apical. Now this is where the 2 is. And at the apical membrane, then 3 is reabsorption of the organic nutrients and certain ions by co-transport. So this is 3 here, co-transport, and then reabsorption of water by osmosis through the aquaporins. That's number 4 here. Aquaporins are these special channel proteins, beautiful pink colored ones, in which you can see how they will be. They're especially for water. So aquaporins, reabsorption left behind, and then of course the solutes can then be reabsorbed as they move down their gradients. Then number 5 is the lipid soluble substances diffused by the transcellular route, and number 6 is the various ions, chloride, sodium, calcium, and urea diffused by the paracellular route. So beautiful diagram, very well explained. I would like you to pause it here and have a look at it. What is primary active transport? What is the secondary active transport? And secondary active transport means it is in response to the main active transport. So it's just like the teacher walks out of the class and then you all walk out of the class. So basically, if the teacher didn't walk out of the class, the biology class is finishing. So Dr. Saira walks out of the class and then you, of course, all follow. So the primary active transport and then the secondary active transport, passive diffusion, transport proteins, ion channels, aquaporins. And these are, of course, beautifully explained in it. And we go back to the question now. So, I'm not writing the answers on the page. I'm just adhering to the mark scheme and I'm going to explain this. So you've got the microvilli and the basal membrane. So the microvilli are this, which is towards the lumen side. And the basal membrane, of course, are these ones. These are the basal channels or the basal membrane fold. So these are the basal membrane. This is the lumen side, and this is the basal side, which is the outside of it. And this, of course, would be the entire cell. This would be the lumen inside, and these would be the ones on the outside. So microvilli increase the surface area. That's the first point. Then for many transport proteins, why? Because you have the sodium potassium pump here, and then you have the co-transport channel proteins here. So you have a lot of things which have got to be stick into this phospholipid bilayer. So you've got the co-transport channel proteins on this side. So many transport proteins, then sodium and glucose are co-transported into the cell. Make it very simple. Sodium ions and glucose co-transported into the cell. Do not make it difficult for yourself. There's a habit which you all don't know how to really, this is called smart studying. Then sodium ions pumped out of the cell into the blood. So sodium ions are going to be pumped out of the cell into the blood by the sodium potassium pump. And then active transport, and then many mitochondria. Each cell of this has many mitochondria in it. It has, every cell has mitochondria, but these have many mitochondria. Why? For the energy or for the ATP. And then tight junctions. Why? The tight junctions are between this one and this one. So this is the tight junction here. Tight junctions are to prevent migration of proteins. Why? Because the channel proteins should not move, to prevent migration of channel proteins. Why? Because this sodium potassium pump, which is on this side, must not in any way sort of, you see, phospholipid bilayer, lateral movement. What if it goes this side? No, that's not the wrong side. So there should be prevents migration of channel proteins. So that is why the tight junctions are between this cell and this cell. So there is this tight junction here and this tight junction here. So it is going to prevent that the phospholipid bilayer, which allows lateral movement, the channel proteins do not sort of migrate to the other side. They should not migrate to the lumen side, or the ones which are on the lumen side should not migrate to the basal side. So that was there are about 7 points, and you had to give me any 5 to get your 5 out of 5. So what is the intelligent way? You just write a lot. Write more so that, one or two maybe is purely wrongly worded and you get your 5 out of 5. Then let's read through question number part C. Figure 1.2 shows the concentration of ADH in the blood at different percentage changes in the water potential of the blood. Now you can see here there is -20, -10, and then 0, and then +10. So the highest water potential of distilled water, the highest water potential is 0. And you add, say, 10 solute to it, it becomes -10. You add more solute, it becomes -20. And then, of course, you add pressure to it so that it becomes +10. So here you can see there's +10, this is 0, which is the highest water potential, and then this is -10. So here the water potential is decreasing because 0 is that of distilled water, and that is the highest water potential. So -10 is less, -20 is even more less. So it's more negative, less negative. That story which we do in the AS level. And +10 would, of course, be increasing by pressure. By exerting some sort of pressure, the water molecules will be forced to move in the direction of the pressure which is being applied. So they've given you this. Please understand the graph. And what you have on this side is the concentration of ADH in arbitrary units. So you had 0 here. I'm just going to clear the page. And so you had 0 here, then you have 2 here, then you have 4 here, and then 6. So concentration of ADH is increasing. Now, as the concentration was somewhere near 15 or 14 point something, and it was -20. Then it is -10, it is decreasing. Then the concentration of ADH is decreasing. So -10, it is decreasing. -20, it was here 14 or 14.5. -10, it is here 8 something. 0, it is 2. So there's less ADH. And then the ADH sort of remains the same. It is 0, it remains at 2, 2 here, and then it becomes sort of constant. So always read this graph just like I'm reading this graph to myself. Read this graph to yourself and then go on to the next question. It says describe the trend shown. Now describe, what is describe? Describe is no biology. Describe is no biology. So no biology means anybody, any child who has not even studied anything can do this question. So these are the two questions which you'll probably get if you did not study any biology. It will be sad if that is the case. So describe the trend shown in figure 1.2. So the trend would be as water potential increases, concentration of ADH decreases, which you can see, of course, as it is decreasing. So as the concentration, as the water potential increases, the concentration of ADH decreases. Between 0% and 10%, the ADH concentration remains the same. And then two figures. So at minus 20% percentage change in the water potential of blood, please write this whole thing on the x-axis. The ADH concentration is in arbitrary units 14.8, and at 20 minus 20% and then minus 10%. Sorry, I made a mistake here. So minus 10%. So minus 10% is this one here. So the is 8.4. I might be a little wrong, so please correct me if I'm wrong. Coming on to part two of the question, you can see sometimes a person will have a low concentration of ADH in the blood, even though there's a change in the water potential. Suggest one effect. Now, I always say suggest are the ones which actually discriminate between the A star and the A student. So if you know the suggest, you're probably going to get a few extra points, because suggest is always very difficult. And I often say just leave the suggest part, because it is something which is very challenging. And if you waste a lot of time on it and you are unable to do it, then it's really futile. So suggest one effect on the circulatory system of a low concentration of ADH in the blood. Now, so of course, if ADH is not going to be there, then no water is going to be reabsorbed in the DCT and in the collecting duct. So no water reabsorbed. That means the blood volume is going to decrease, and this, of course, will result in pressure will decrease as well. Because as the water is absorbed, the volume is restored. Like if you sweat on a day, on a very hot day, you sweat a lot, well, then of course ADH should be released, because more water should be absorbed from the urine. So there should be less water in the urine. So of course, if that is not going to happen, then the blood volume is going to decrease, and this is going to decrease the pressure as well. Now let's look at question number 2, which is cotton and false flax. I don't read the biological names because that makes me more nervous. Are crop plants that are grown in different parts of the world. Rubisco activates as an enzyme, activase is an enzyme in the stroma of the chloroplast that is needed to maintain the activity of a second enzyme, Rubisco. So, I mean, immediately you should be thinking of the Calvin cycle and Rubisco, which I'm just going to revise with you. Scientists measure the activity of Rubisco activase in cotton and in false flax at a range of temperatures. So we took a range of temperatures, different temperatures, and we measured the activity of Rubisco activase. Rubisco activase activity in arbitrary units, and then of course I will use different colors. So this is cotton. So let's look at the graph for cotton, and I see it increasing and then decreasing. And then let's look at the false flags, and let's look at that in a separate color. So the false flags increases and then decreases, and this. So now whenever they give you a graph which sort of intersects each other, there is a very important point, because they want you to be picking up this point, which is the point of intersection, and they want you to be talking about that. Remember that. That's always their intention of Cambridge. So what do we have here? We have different temperatures here on the x-axis, and here we have the Rubisco activase activity. So it's the activity of an enzyme, which is a very typical graph of temperature and enzyme activity. So understand it and sort of spend a little time on it. With reference to figure 2.1, compare the results. So compare is always means two things. It means similarities. So what are the similarities here? And number two, the differences. So we're going to be talking of the similarities and differences. So compare doesn't mean only differences. Compare means, okay, you compare two things. So with reference to figure 2.1, compare the results obtained. Now that's again a very, very easy question. And I mean, there's no question that anybody should lose marks on that. And that's a very good, how many marks is 4 marks. So it's a very good 4 marks, easy to get 4 marks. Now, I'm going to just discuss the mark scheme with you in a minute. Now, everybody should be very clear when we are talking of similarities is that there's something which is common to both of them. And you can see both of them increase, then decrease. So you see this increases, then decreases, and similarly, this increases and then decreases. So the similarity is both activity of both in both cotton and flax. Even if you didn't write in both, you didn't write their names, and if you just said in both increases then decreases as the temperature increases. You see, this way the temperature is increasing. The other similarity is that activity is the same at 0.104 AU, and this is the point which they are talking about. This is the point that they are talking about. This is the point that they are talking about. 0.104 AU, 0.104 AU. So this is the point where it intersects, and this is at 30 degrees Celsius. At 30 degrees Celsius. That was the two similarities. That's a very, very important thing to remember. Whenever they show you two graphs intersecting each other, you have to, the point where they intersect, with the point where they sort of cross each other, that is the point that they're talking, and that means the activity is the same. At 30 degrees centigrade, the activity for cotton is also the same, and the cotton then for flax is also the same. Okay, now let's look at the differences. Now, this were the similarities. Now, let's look at the differences. Now, when you look at the differences, let's look at the differences now. The first differences which I'm going to talk about is cotton has a higher optimum temperature. Why are we saying cotton has a higher optimum temperature? Because what is cotton? Cotton has a higher optimum temperature. You see, this is the optimum temperature for cotton. This is the optimum temperature for cotton. What is the optimum temperature for flax? That is somewhere here. So 25 and 30. So cotton has a higher optimum temperature. Then the highest peak activity is higher in flax than in cotton. Activity is higher in flax, is 0.12. Here it is somewhere here 0.10. So second point is maximum activity or higher activity in flax than in cotton. The third thing is cotton can work at higher temperatures than flax. Now, if you look at cotton, cotton can work at higher temperatures. You see, this is now where it's still working at higher temperatures, while here this has finished. While here it is still working. Now look at this temperature from here to here, it's still working. So cotton can work at higher temperatures than flax. Then the fourth mark scheme point is that more than 30, flax has a higher activity. After 30, if you look at it, flax has a higher activity. Look at this graph. The graph is, this is the graph. So there's higher activity after 30. So if you look at less than 30, now what is a little different? If you look at less than 30, flax has a higher activity than cotton. Less than 30, flax has a higher activity than cotton. Please keep on looking at the graph. And more than 30, cotton has a higher activity. So more than 30, cotton has a higher activity. And so these were the differences that we were seeing. And you can see the differences were very visible on the graph. And I'm not going to give them in writing because I'm sure you all can pick up the mark scheme and have a look at it. But I've explained the points to you. So then we go on to the next part of this question. Now, suggest reasons for the differences. Now, this is, of course, something which is a lot of biology involved. Suggest reasons for the differences shown in figure 2.1. Now, I mean, why would they be asking you reasons for the differences shown in figure 2.1? But if you look at this graph, which we've been talking about earlier, in this graph, what are we studying? We're studying enzyme. Now, when we're studying enzymes, what are we looking at? We're looking at how would this be different in different this thing? Why would that enzyme be different? Well, of course, in cotton is adapted to live in areas with higher temperatures. Okay, why? Why? Because it's a different alleles, different primary structure of the enzyme Rubisco activase, so different tertiary structure. So different tertiary structure of the Rubisco activase. That is why it is more resistant and it can survive higher temperatures. Like just we have, if you remember in the chapter on genetic engineering, we talk of Taq polymerase. And Taq polymerase is a DNA polymerase, but it's found in bacteria. So how is that different from the DNA polymerase in your body cells and my body cells? There has to be something different. The primary structure would be different. So that means the tertiary structure would be different. And so it would not be affected by high temperatures. The hydrogen bonds would not break, or maybe it does not have many hydrogen bonds. So that is why it is more resistant to high temperatures. So always understand when they're asking about an enzyme, they are asking you to refer to its the allele for it, or the gene, you could even have used the word gene for it. So different alleles, different primary structure, and so different tertiary structure. Then coming on to the B part of the question, it says Rubisco enzymes from cotton and flax are active at temperatures up to 45 and will denature at 45. So they will denature at 45. Explain how the Calvin cycle is affected when Rubisco denatures. So now they're asking you to how the Calvin cycle would be affected. So I mean, let's for a minute just go through the Calvin cycle. But if you look at the Calvin cycle, what is going to be happening is in the Calvin cycle, basically what is going to happen is there's going to be no CO2 fixation. So there's going to be no CO2 fixation, right? No CO2 fixation, no GP made into TP, no regeneration of RUBP, and no glucose made. So very simple, Calvin cycle is not going to happen, no complex fixation, no GP to TP, no regeneration of RUBP, no glucose made. Okay, then we come on to the C part of the question. One goal of genetic engineering is to make crops that are heat tolerant. This means that crops can grow and produce a good yield at high environmental temperatures. I use the information given in question 2 to suggest and explain a way to improve the tolerance of a crop to high temperatures. So, very simple answer to that was we take the gene for the Rubisco activase because it said one goal of genetic engineering. So use the information question 2 to suggest and explain. So they asked you to talk about the gene which we talked about earlier on. So we talked about the Rubisco activase. So take the gene for Rubisco activase from cotton and insert it into embryo of whatever crop that you want to, whether you want to do it into the flax or into any other crop. And so that you can maintain the action of Rubisco at high temperatures, which is of course more than 37.5 and up to 42.5. So this was the genetic technology which was mixed with this chapter question. So whenever it says gene technology, well, you have to realize it's got to be something to do with taking a gene of interest and putting it into something so that we can then have the protein or the product which the gene is going to be dictating. So that protein would be made in that other organism in which we are putting it. So if we want a crop to be surviving at high temperatures, well, we'll have to take this Rubisco activase because basically it's the entire process of photosynthesis which has to take place at higher temperatures. So I end this here now and we'll continue with the rest of the questions in the next video. And thank you very much, and please do subscribe and do comment if there is anything else which you would like me to do. Thank you very much.